your email is going to talk about the like what week you're going to be in. When I'm, to divide up the class here, so we'll have like an in-class session for a particular group of people on one week, and then during that same week we'll have a crowd online, and then the following week everything will switch. But I would like to say that this does not mean that you are stuck being online only for some of these weeks. If you would prefer to be online every week, or sorry, in person every week, then please reach out. Let me know, and I'll see what I can do. I realize that some people learn better in a more kind of in-person situation. I know that I'm one of those people. I like to be more in person. But that doesn't mean that that I mean, when because you've been put down for an online week. It doesn't mean that you have to stay online. So reach out, let me know, and I'll see what I can do. But I just like to make sure that there's enough seats in that room, which are separated by 6 feet from the next seat. And this is not my rule. This is the campus's rule. So please bear with me, and I'll see what I can do to accommodate. Otherwise, but just please check your mail. I'll also send out the message by Slack. Basically, what I'll do is that once 5 o'clock on today, or even tomorrow morning, once the ad drop crowd has settled to where they are, and the numbers in the class have stabilized, then I'll make those lists, and that's why there's a wait. But next week is going to be in person. They will always be. I'll always be on Zoom, so you can always log in here. So I know that there are some of our colleagues in this course are actually not in Meadville, and so you don't have to stress and get here. You can still log in by Zoom as you like. Anyway, another thing I wanted to talk about was come with me to the website. There we go. On the website, I've added a new thing. This is if you go if you go to cs.allegheny.edu and then you just click on my picture there, you'll find this site here, and then you go to classes, and then you go down to Bioinformatics. This address is also in the syllabus. If you scroll down, where is it? Here. I'm going to start adding all of the videos that we cover in this course on YouTube. That means we have a YouTube channel, which is actually quite exciting. I think it's exciting. But we have a YouTube channel, and so what that means is that the videos that or the videos from our classes are going to be on YouTube, and so you can reach those if something happens where you're unable to come to class. This is partly because of the pandemic, but also though, when I was going through my classes, it wasn't so long ago that I was a teacher, that I was a student, before I was a teacher, some of my classes were on YouTube, and I found that incredibly helpful to learning because, for instance, I'd be in class, I'd be writing things down, and then I always found that while I was writing something down, or if my computer crashed or something happened, I would miss the stuff that happens in the classroom, and that was really, really annoying. But now you don't have to miss anything because you can just find all this stuff on the YouTube channel. So you can click on, for instance, class videos. I'll just go ahead and make this and open this in a new link here. And this is what the channel looks like, and we have Bioinformatics as a channel, a playlist right here. These are some of my other videos from other classes. You can always look at these things and see what you think. The classes are databases and analytics. They're interesting classes. You can always have a look at those if you want. And if I have other videos, I'll just put them up there too. This GNext, this is a project in case you're wondering. This is, you can have a look at my research that I put together over the last, I think it's like 4 months. You can kind of see where my interests are in data analytics and bioinformatics. GNext is a gene expression analyzing software. It's still in development, but here's my presentation that discusses it for a conference that I went to, or that I got accepted into fairly recently, which is quite exciting. Anyway, but I'm still developing GNext, so there's probably some things you can think of that are probably it's not exactly perfect. Anyway, so there's that. And so I do hope, though, that when you are kind of preparing for exams in this course, or if you miss a day or something terrible happens where you just cannot be here, you do have your YouTube channels. Now, It may take me some time to get all the videos up. Like for instance, I'll try and have them like done by the next day, but certainly by the next during that week I'll have the videos up. It does take some time to, if you can believe it, to render these videos. It takes on my machine actually quite a long time to take the video from class and then to crunch it and kind of compress it and do what it has to be done before I can send it to YouTube, and then YouTube then has to do some other stuff, and so it does take some time. So I will try and get these videos up there promptly, but if they're not, then just keep checking, and they should be there soon. Okay, so I think that's really all I wanted to talk about there. If you scroll down to the bottom of the, oh, by the way, if there's any questions, please let me know. Just let me know. Just start, open up your mic and start speaking. And the reason why I say that is because I don't necessarily have the chat open on my screen, because I have just all this other stuff on my screen. So there really isn't much screen real estate. And so it's just kind of a mess. So if I don't see your chat, you can still send a message by chat if you'd like. But if I don't see the chat on Zoom, then just start talking and I'll hear you. Okay? So on that, are there any questions? Okay, so let's get back to where we were the time before. This is from last Tuesday. We're still in the introduction, and we're going to finish these slides today. Then we have an activity at the end of class. And so it should be actually quite exciting. I'm looking forward to today's class. Let me make sure I have my notes ready for me. Good. All right, so where were we before? We were talking about, I guess I'll start here, because this is probably one of the most important things. But we were talking about, like, what is bioinformatics? And one thing that we mentioned last Tuesday was that bioinformatics seems to mean a lot of different things to many different people. And that's perfectly legitimate here. That happens a lot with some of these interdisciplinary fields of study. Bioinformatics, as you remember, is a field of study which is made up of computer science, biology, and mathematical sciences, like statistics. And so you have people and ideas coming to this area from all these different areas. And so they've really kind of constructed parts of the discipline which look very much like mathematics, or very much like biology, or look very much like computer science. And so that's why this picture here is so relevant, where you have these people who are blindfolded touching an elephant, and each person who's touching this elephant, they're touching a different part of this elephant. I still feel sorry for this elephant, though. We shouldn't be abusing this elephant in such a way. But when they touch this elephant in different areas, they think, oh, wait, I'm touching a tree trunk, the person says, touching the elephant's leg. And someone else is touching the elephant's tail, and they said, oh, I'm touching a vine. And then someone else is touching the elephant's nose or the snout, is saying, oh, I'm touching a snake. And so the thing is, though, that it feels very different depending on what kind of area of bioinformatics you're in. But don't worry about whether you're in. I mean, if what you're doing in bioinformatics does not look like what your neighbor is doing in bioinformatics, that doesn't mean that you're in some different type of discipline, but rather you're just working on a different area with a different type of concentration. I think I mentioned that when I was in a lab in graduate school in bioinformatics, there was a woman who was next to me, and she was working on some very sophisticated looking gene and protein networks, which I thought was absolutely fantastic. I mean, just very exciting stuff. But I wasn't working on that. I was looking just at actual words in DNA. We'll talk about that in this class, actually, which is actually quite exciting. But I remember I was thinking to myself, I was becoming kind of self-conscious, like, well, wait, am I really in bioinformatics? Is this a different area? Am I not doing the same stuff that she's doing? Because surely if I'm in the same area, then we would be working on projects which are very, very similar. And it doesn't work that way. But what I'm trying to say, though, is bioinformatics has many different approaches, and there's room for everybody in bioinformatics, whether you're interested in the biology, or you're interested in the mathematical sciences, or you're interested in the computer science. And so there are many different ways to approach it, which I think is actually really refreshing, because you can play to your strengths. If you're more of a computer science person, then you can be very, very successful in bioinformatics. In that way, or if you're good at biology, then you can be very good at bioinformatics and biology. And so that being said, it means a lot of different things. The field of bioinformatics means a lot of different things to many different people. But I think, though, that one thing that we discussed on Tuesday is that while these questions in these different colored blocks are actually they're different types of pursuits in research. That is true. There are different pursuits in research, but if you study kind of like the overall kind of image of each of these questions here, the overall kind of the approach, you'll see that actually they're all using DNA or genetic material in some way to answer questions. So, for instance, the first one here is the science of collecting and analyzing complex biological data, such as genetic codes. Again, it's a research that's gathering around collecting DNA samples, whether that means you're analyzing them or whether you're just collecting them. It's still bioinformatics. Here's another one here where the field of exploration into data to describe the onset of illness, disease, and medical disorders. Again, you're looking at DNA. And the interesting thing about bioinformatics, which I think that when you first see this, it blows your hair back and gives you goosebumps and makes you feel really excited. And that is that when you look at DNA, DNA is actually just a kind of a long string of characters. It's made up of an alphabet of 4 different bases, A, T's, G's, and C's. And in that DNA, you find the genes, and some of those genes have typos, typographical errors. And those typographical errors, misspellings, if you will, are the things that cause certain types of gene breakdowns. So, for instance, you can imagine that DNA is kind of like a text, a paper that you may have written, and if you have a typo in there, then it kind of works against the coolness or the enthusiasm of the paper. Whereas, for instance, if you have typos in DNA, sometimes the repercussions of that are actual genetic disorders. So if you're studying disease and medical disorders in DNA, again, you're looking to find where those typos are. And then there's something called gene therapy, which is the study of trying to, I have to use this in a kind of a computer science term here, but you're upgrading or you're updating the firmware in some genetic system. In other words, you're trying to fix that gene by correcting the spelling mistake. So we will talk about that. It's actually a very cool area. It's there's a lot of work to be done, and some of the work is actually quite exciting. Let's see. For instance, study of living. There's another one here. This is actually a similar idea here. The study of living systems to determine patterns of life and health. Again, the patterns of life and health may be spelled out in someone's genetic code. For instance, maybe your health gave you excellent red blood cells, which are able to, or maybe your genetics gave you excellent red blood cells, which have an extra ability, if you will, to hold on to an oxygen molecule in your, it builds a protein which is able to better hold oxygen. That might make you able to hold your breath longer. Maybe that's a trait which came because your descendants were living in the mountains. And this is true. If you go to certain places in the world where you find populations that tend to live in the mountains or extreme climates, extreme environments, you find that their ability to create red blood cells to hold on to oxygen is better than people who have traditionally lived or organisms, I should say, organisms who have traditionally lived at sea level. Because at sea level, it's not necessary for survival to be able to hold on to as much oxygen as you possibly can. However, if you're living in the mountains, if you're an organism that lives in the mountains or a person, then it's to your direct advantage to be able to breathe up there. So yes, and so there are certain types of patterns that you can see from the genetic code. You can say, I can see that this is a gene that produces this protein that creates or that holds on to or that it traps oxygen and holds on to it longer, allowing all the cells in the body to become oxygenated, or maybe you have better, maybe there's a better cell in your lungs, for instance, that pulls more oxygen out of the air. Then maybe that's a pattern that you need to sustain life. And so all of these, that's another study that you'd use in working with DNA. There was something else I was looking at. I forget where it was exactly, but there's, for instance, here, the relatedness. Here we are. A framework used to determine the relatedness between people, dogs, cats, mice, and rabbits and any living thing. This is, again, a study of DNA. You're looking to find the similarity of DNA. I guess the best way to describe this, and we actually do cover this in this class, and we spend a lot of time talking about comparing DNA, but the best way to describe it is that imagine that you're a school teacher, and you suspect two students of cheating. How would you determine whether those students were cheating? Imagine you suspect plagiarism, for instance, where you've taken someone's taken some text from someone else's work and copied it into their own paper, saying this is my paper now. Well, how do we detect plagiarism? We detect plagiarism simply by comparing. We look at the text that's in this paper, and we look at the text that's in this paper, and we find out, is there anything in common? And if it's in common, how similar is it? Are the words exactly the same? Are there any spelling mistakes or some kind of strange ways of describing something, or some strange word usage, which is conserved in both texts? In other words, is the word misspelled over here and it's misspelled over there? How do you detect these things? And so this is a way of determining plagiarism by finding out that these two texts are the same thing. And so, for instance, when you're looking for relatedness between organisms, you're looking at the DNA, which is, again, just a string, and you're determining, well, how much of the DNA in a rabbit can also be found in a cat? You're just looking to see whether there's how much similarity there is. And so that brings me to the next point here. So when we're working in bioinformatics, we start with a gene that looks like this, which is this molecule picture that we see here. It's what the gene will look like when it's coiled up around a chromosome, or in the chromosomes here in your cell. That's what the DNA looks like. But to work with it in bioinformatics, we have to convert that gene into text. And this line this block below, the A's, T's, G's, and C's, this is the text. And so, in other words, this is where all the analysis comes from, just by looking at the A's, T's, G's, and C's here. If you remember, you can actually click on this thing. I'll just take you back in case you weren't in class on Tuesday. If you're new here, welcome. This is a record. Every gene, every protein that we know in science has been written out in this NCBI database. And so you have this. You basically have this kind of it's a rap sheet, I guess, if you will, of a gene. And inside the gene, for instance, and we'll talk about this too, but I'm just giving you kind of a heads up. But you have some very specific information. This is like your serial number, name of a gene. That's how, that's how you know what this particular genome is, which is like a volume, which is a collection of all the gene material. This is how long it is, and then you have some information here about whether it's DNA or whether it's RNA. In this case, it's RNA. We'll discuss what all that means, and then we have another type of physically what kind of sequence is this? Is it a long line, or is it rather a ring that goes round and round? Rings tend to come from the genetics of bacteria, where it's like a big loop of code, if you will, or if it's like a long line, it's this. But it does make a difference to how you approach the study, knowing all these little pieces of information. For instance, this is the size. This is saying that this gene for the Wuhan isolated COVID virus, that means the virus that we discovered in Wuhan. Again, we don't know how it necessarily got there, but we know that it was first found in Wuhan. Let me just say something about that. It's very important that we say that this is the virus which was discovered in Wuhan, rather than using some politically incorrect term to suggest that it belongs to a specific nationality or culture. That's just ethically unwise. That's just not right. So this is the virus which was discovered in Wuhan. And so, again, it wasn't, they suspect that maybe the COVID virus came from bats. Maybe it somehow jumped from bats to people. Again, they're still working on the World Health Organization is putting together some ideas on where it came from, but this is the actual sequence of that virus which was discovered in Wuhan. So anyway, all this research that you can see, though, or this list of names and things here, this is all kind of like the backing for how do we know that we are actually working with a virus, or how do we know this is a virus? How do we know that this is the COVID virus? And so you'll look through here and you'll see like all these different kinds of, here's some authors. These are people who've done research to say that yes, we've sequenced this gene and we are sure that by our comparative studies that this is actually a SARS type virus. And that's where the name comes from. And so if you go through, you'll just see that a lot of work has been done to create this virus. Here's actually kind of interesting here. Here's a paper here that came out in 2005, which is called The Structure of a Rigorously Conserved RNA Element Within the SARS Virus Genome. This, if you will, is kind of like the background information that all this stuff is resting on. The whole notion that this is a virus that comes from SARS, or a relation of SARS, that is all being kind of backed up by this paper here. This is a peer-reviewed paper. That means you can't just publish this thing without actually having many experts in the field read this paper and then confirm that this is good science. But this is a paper, though, that says that we know now what the structure of a SARS virus looks like. And therefore, these researchers up here use this paper and others, which are all listed down in this paper here, but they use the knowledge from these papers to say, yes, we believe that from our comparative study, this was actually a virus which is related to the SARS virus that came out in 2005 and other ones before that, I think. Anyway, we actually do spend some time to go over these records, and so you have an idea about really what everything is. But I'm showing you this record, though, to get back to my earlier point that, I mean, look at all the work that had to go in to actually confirming that there was a virus that was isolated in Wuhan, which is the SARS or COVID virus. All of these people here approached bioinformatics, or they used bioinformatics methods to conduct their work. Their research was all brought to us by technologies in bioinformatics, which was, again, brought in from biology, from computer science, and from the mathematical sciences here. And so all of these people here found a way to contribute to this one project through either bioinformatics or biology or mathematics or computer science. And so what I'm saying is that there's room for everybody. And everybody, I mean, of course, I'm very biased here, but everybody should be very interested in this new area because there's so much stuff that needs to be done. And if we have more people working on these things, then perhaps if we have more people understanding how the coronavirus works, perhaps we could end this. For instance, if we have more people working on how it infects, and more people understanding like how the method is, or how it works, or how it or what goes on at the molecular biology level, or if you can take some data and analyze it and find some trends and patterns, then maybe, for instance, we could stamp out this virus and any other SARS virus, and perhaps even continue this motivation and an approach to research and stamp out other types of viruses which work in similar ways. And so there is a lot of work to be done in bioinformatics, and there's a lot of room for everybody to be there. And so this brings me to my next point, getting back to the slides here. But how much data actually is there to study? We'd be surprised. This is a graph here, which is talking about the growth of something called GeneBank. Now, GeneBank is it's a giant online, free, publicly accessible database that stores sequences of DNA. So that means that if you're doing some kind of research and you isolate some kind of a gene from an organism that you're working on, you are highly recommended, highly recommended here to submit that gene that you have sequenced. In other words, you've started with this genetic molecule here, and then you put that genetic molecule into this text of A's, T's, G's, and C's. Then you're really asked by your community to submit it to GeneBank, which is this giant database, which is where you can find seemingly all genetic knowledge known to people. It's all right there, GeneBank. Everything is publicly available. And the reason for this is because if everyone starts keeping secrets and not letting anyone know about the sequences that they find when working with certain types of genes or proteins or whatever they are, whatever you're looking at, then research will come to a halt. And so there's a kind of a consensus that if anybody finds a protein sequence, or they take a protein sequence and then they figure out what the gene is that makes that protein, then they are to put that knowledge into GeneBank so that other people can profit from that. Not profit from financially, but so that research can continue. This reminds me, actually, back in the early days, when genes were first being put into this GeneBank site, there were a number of organizations that were trying to prevent that from happening. They were trying to make people, they're trying to like basically buy all these sequences, and if anyone did any research with a sequence, then they had to pay royalties to some group that held onto these sequences. That was stamped out after a lot of litigation in courts, and the argument was that we all live in Pennsylvania, and so we're all allowed to draw the state of Pennsylvania on a piece of paper and not have to pay anybody to draw that state. And so the idea is that why should you have to pay anybody? The map makers of Pennsylvania. I live in Pennsylvania, but why should we have to pay anybody to draw or work with our own map of Pennsylvania? And so it's just it was decided that it wasn't just to make people pay for these things. And so the same idea goes for genetic code, and that is that it's kind of a map of genes, and so that if you have that, or if you're working with that gene, you don't necessarily have to go back to the cartographers or the map makers of those genes and pay them royalties, because that's just it's just unethical. It's just not correct. But there was a time though when people were thinking that in order to be a bioinformatician, you would have to have very, very deep pockets to pay for the genes that you use, because they were all copyrighted and whatever. But that's not true. That's all been found out to be not true. And so you can do any research you like with genes. Okay. So speaking about research with genes, where did some of this research data come from? And so here's some notable examples I wanted just to cover with you. And there's something called the Human Genome Project. This was something that was going on from like the late 90s to about the early 2000s. It went on for a long, long time. And the whole object of this study was to go through all the human chromosomes and to convert all the genetic content in humans into A's, T's, G's, and C's. In other words, to write down everything that you have that you see in the molecule into a giant text file. For each chromosome, they wanted to have all the genetic code. There were 5 different people that were chosen. I think that Steve Jobs of Apple was one of them. I'm not sure about that, but I think he was. There was someone else called Craig Venter, who was he's still kind of like one of the high flyers or the big shots in bioinformatics research, but he has like a bunch of companies that do bioinformatics based research. And so they used a drop of their blood, basically, and ran it through these machines called sequencers, where they were figuring out what the DNA sequence actually is. Like, if I were to start at the very, very beginning and write down all the A's, the T's, the G's, and C's, what would that look like at the very end of my task? And there were all these notions that began in this project where like they were thinking, oh, well, we're humans, and so therefore we must have this really evolved, crazy evolved genetic code here, which is going to be completely different from anybody else. Actually, it wasn't that different from even like bacteria, which is kind of ironic. That actually were very similar to seemingly all biology on the planet. I guess we all knew that, but our vanity got in the way and made everyone think that we were somehow, angels evolving or something. But actually, though, one of the most important notions here is that after this project began, a lot of other projects began where they wanted to sequence like rabbit DNA and dog DNA and frog DNA and deer DNA and all these other types of animals that you wouldn't necessarily think are going to be sequenced. But they ran all these different projects from the technology that was developed for the Human Genome Project. And so now we have seemingly, well, not all the DNA that's available, but we have a lot of DNA to work with. And so if you're working in, for instance, rabbit genes, then, for instance, you can look at, go back to GeneBank, and you can find the genes that rabbits use to live. And so this large-scale project, actually, to determine the sequence of human DNA, is probably one of the big monuments in bioinformatics, because it created so much technology to figure out what the genes, what they look like. This is a link, by the way. If you click on this anniversary over here, you can actually, there's some more reading about this. You can kind of like find out about the 20th anniversary landmark of the Human Genome Project. If you scroll down, there are some interesting videos, which I encourage you to look at. They're all quite exciting. In fact, this interview right here with Eric D. Green, MD, PhD, he was one of the organizers of this giant project, and he had some very interesting things to say about how stressful it was to work on this thing, yet how exciting it was at the same time, and they were all making history, anyway, it's one of those cool, tell me more type articles. It ends too soon. It's an interview, but it ends too soon, I think. Another important study that is more recent that uses the same types of technology that comes from the Human Genome Project was, now we know how to take a piece of protein, basically, or some organism's organ, and extract the DNA. That means to go into that organ and to look at the actual ATGC makeup of that organ. And so this is just stunning, actually, this article. This is something that was fairly recent. You can click on this link that's in here, the oldest DNA. But basically what this is a Washington Post article, and there's a lot of other work in there too. You can find some other background information there too. But there's a bunch of articles that exist on this topic. But researchers found mammoth molars. These are the teeth in mammoth. These giant prehistoric, now extinct, elephant-like creatures that roamed the earth, which were just like Jurassic in size. They found these perfectly conserved molars, and they were able to extract the DNA from the molars, which is magic if you think about it. To find the ancient DNA of some organism. So the big question here is, it gets into ethics here, but they're actually saying that now that they've extracted, let's see, scientists extract the Siberian mammoth molars. This is the oldest DNA ever discovered. The big question now is, well, can they use it? Could they somehow use this DNA, put it into a cell, and then to bring these mammoths back from extinction? It's getting there. We're getting close. We're getting close to doing that. But FYI, one of the problems with this ancient DNA is the same problem that perhaps you and I may encounter if we pick up a copy of Shakespeare and just read this before going to bed. I mean, it's complicated reading. The text is very, very different. The way that the language is used is very, very different from the way that perhaps you and I would speak. And the vocabulary is different. The makeup of sentences, the inversion that they use. If you look at Shakespearean text and text from today, it's very different. It's very, very different. And so the DNA also registers the same types of dialects and the same types of differences. And so if you're looking at ancient DNA, it's true that the genes are there. But if you're trying to read those genes and understand them with the same lens that you use to understand genes from today that have been evolving for like the last 1,000 years, 2,000, 3,000 million years, whatever it is. I mean, these are like really, really a long time ago. But if you're looking at these genes, you'll see that there's actually a different voice, if you will, that's speaking that sounds the Siberian mammoth genes from the voice that sounds the human genes or the rabbit genes of today. Evolution has taken its course, and so the DNA is now different. And so many of the studies that you find are talking about how do they bridge that gap? How can we take the genes or the woolly mammoth DNA and recreate these mammoths? One of the problems that they encounter, though, is that the genes or the sequences are broken in some places where the code just wasn't able to be read. And so they have to superimpose data or DNA from more contemporary organisms into the genome for an older organism. Now, you might think, well, that's fine. You just splice this stuff together and it's all good to go, right? It's like just cutting up, pieces of tape and like kind of make a cut in the middle of the tape, and then you kind of pull everything apart, and then you superimpose the new piece of tape, and then you kind of, it's like you're editing movies. It's not the same because there's a dynamic that exists in DNA where, for instance, a gene that's or some piece of code that's listed maybe back some distance actually has implications and repercussions to genes which are forward or downstream of that gene. So, for instance, it seems that you're not just reading like a Turing machine. You're not just reading DNA like at the place where you are, and then you move down to the next area, and then you're done with all the stuff that works behind you. Rather, some of the ideas are that you're creating proteins which are sounded down here, or they begin down here, and then as the protein or as the new proteins are being developed, then something that's made before has to somehow come back again and fix something that happens. So there's all this interplay between seemingly all these different areas of DNA. That's actually a study. That's actually an area of research where they're trying to. It's kind of, you use a lot of molecular biology, but once that molecular biology has extracted the actual DNA code, then they use automation to try and figure out how does this section of code interfere or enhance or somehow interact with a section of code that's well back here. Upstream and downstream DNA appears to have some kind of connection. And when you're working on this kind of research here, the only thing you can really do is just run experiments. There's no manual to read. It's not like you can go back and read. If you're working with software, for instance, there's no documentation to read about how this piece of software in the beginning interferes or works with a piece of software at the end in the code. But rather in DNA, you just have to kind of figure out these things. So I say that because when you're doing that research, it is about finding the I don't know. It's about kind of having the creativity to make or to invent or design an experiment which would be able to tell you whether this piece of code over here is interfering with this code over here. How do you design an experiment that does that? And so for that reason, there are many projects in bioinformatics that are just waiting to be solved. And if you have the time and you're interested in the challenge, then go for it. You might find that you've become the world's foremost expert on a gene which came out of a Siberian mammoth millions of years ago, and you'll find out that this gene actually interacts with some other gene. And that is a paper and something that's just amazing. I mean, it's a real thing. It's a real piece of research you can do. I'm going to say something here. So we use DNA here to answer questions, and those questions are basically, in essence here, the questions are saying how different is the DNA that I have in my hands different from the DNA that I know is true. So what that means is, like, I know, for instance, a sequence of DNA, and then I'll say to myself, how do I know that the sequence of DNA that I know is true? I've done the research. I know that I know it backwards and forwards. How do I know, how different is the DNA that I have that I'm working with from the DNA that I know? And that is actually a research project. And that difference will tell you whether it's the same organism or a different organism that you're working with. So you're really comparing apples to oranges in this case here. You say, I am holding an apple in my hand, and someone just gave me an orange, and I'm going to make some observations and see how these two things are different. The apple is green, and the orange is orange. And so I'm going to say that they're different fruit because they're different colors. Or, for instance, they taste different, or the amount of sugar is different. I'm making some comparison. And so it's only from making those comparisons that you know that an orange is not an apple, because you know what an apple is. This is, you've been doing research all your life on apples, and so you know what apples are. And then when someone gives you an orange, you say, this is not an apple. This is something different. And therefore, this is a different organism or a different species or a different disease or a different virus. And so you're comparing things. But like that expression, it's comparing apples to oranges. Every time I hear that, I always roll my eyes, because at the absolute center core of apples and oranges is DNA. And there's a lot of DNA in common between apples and oranges. For instance, apples and oranges are made out of cells. And those cells are constructed in the same kinds of ways, the same kinds of cells, these eukaryote cells that are in there. And they have the same genes to create the mitochondria and the Golgi apparatus and all these other, and the nucleus and all these things. Those genes are the same. And so when someone says to you, you're comparing apples to oranges, you can say, that's okay. It's got the same DNA. I'm allowed to compare apples to oranges here because it actually works out very, very well. So what I'm trying to say, though, is that although we go around thinking that everything is different and apples are not oranges and oranges are not apples, and we kind of get hung up as people here saying that this is different and this is weird because it's not the same thing anymore and blah, blah, blah. Actually, just because it looks different biologically doesn't mean that it is different. In fact, there's many more things in common between apples and oranges than there are differences. And so there's a lot of being the same kind of genetic material, there's a lot of ways of actually comparing these two fruits. So how do we compare these things? How do we compare proteins, genes, organisms? It's going back to the DNA. You could find the DNA, and believe it or not, and this is something that really you may not see it now, but when you're doing your research, it'll hit you like, wait a minute, all biology uses the same language to describe its genes, the same language to describe its proteins. Incredible. It's like there's this common language. We as people are always thinking that, oh, I haven't been to Germany, I don't speak German, or I haven't been to Italy, I just don't speak Italy, or it's Italian, or my French isn't so good, so I really can't go to France, and I'm really stuck here. I can't speak to these people. I can't make friends there. We think that, but that's wrong, because we can. We can learn the languages here. But the thing is, though, that with DNA, we are learning the language in apples, and that language is the same in oranges, and it's the same in frogs, and it's the same in you, and it's the same as me. And so therefore, anything that we learn may actually be able to be, is knowledge that we may actually be able to apply to other areas. And many times it is. We will do some studies in this class where we look at the proteins which exist in, I think it's different types of cats compared to different types of people and rats and other stuff. And you'll be surprised that there is so much in common with these two, of these animals. For instance, rats and cats, they have the same kinds of fur. That fur has to be made out of protein. The protein is usually the same. Go back to the genes. The proteins are made by the same types of genes. And we have hair on our heads, and it's very similar to the way that animals make their fur. So therefore, we are not so different from these animals genetically. So anyway, let's get back to the comparing part here. So we are comparing our... by the way, are there any questions at all? Does anyone have any questions? I don't want to just keep going on if you have questions. Please do let me know. So we're comparing regions here. Now, one thing that's that may be different, or maybe a big difference in comparing sequences here, is, for instance, here I have a dog DNA. I know what the genes are. I know where everything is. But you give me a new piece of sequence here, a new sequence of DNA, and I want to see how, or maybe I want to try and guess what that new sequence is. And so I look at the sequence and I go through it and I find all these. I find the D's, the A's and T's, G's and C's. I find the DNA. And then I compare the DNA, which I know to be dog, with the DNA that comes from the sequence. And I start uncovering genes, which are the same between this dog and sequence. That could tell me something about what that sequence is. And so this is really getting back to the virus which was discovered at Wuhan, the coronavirus that we call. That virus was... They uncovered some new organism just like this, but in the case of dog, it wasn't dog. They were using another SARS virus, which they knew to be SARS, and they are comparing these two different genomes, or these two different strands of DNA, and they say, it's funny, this unusual piece of DNA that I've just found, this thing that's this new DNA, this new organism that's responsible for this respiratory disorder. Appears to be something that I've seen before. Oh, really? What is it then? What have you seen before? So, well, I last saw these same genes when they were a part of a SARS virus that we discovered in 2004. And so then that tells you something about what that organism is, because you're comparing, going back and forth, back and forth, back and forth, and you say, while the sequences are not exactly the same, there is still enough in these things to make me believe that this is a variant of the SARS virus. And so in the news now, when you see that in California, and my heart goes out to all these people who've had the coronavirus. But in California, for instance, back I think a couple of months ago, they began discovering what they call variants of the virus. What that means is that the virus is in the midst of evolving. It's becoming different. It's a new SARS virus. The original SARS virus evolved into what we call the coronavirus. And now the coronavirus is evolving into maybe some other virus, the COVID-19 DF1 virus or whatever, some other virus. But the thing is, though, that it's still the same virus. There's a lot of stuff that's still the same, but there are specific, maybe if you think of specific changes to types of genes which have gone under some kind of change. They've received a change. And with the California variant that they think is like 70% more infectious or more transmissible, they're saying that the virus has actually received some kind of an alteration here where the proteins on the outside of the ball that contains the DNA or the RNA from the virus, those proteins on the outside have the proteins that create those things, or the genetic code that creates those kind of like sticky things that extrude from the sac of the virus. Those have undergone some kind of modification. They don't modify themselves. They're modified randomly and by environmental pressures. If those random changes become more of an advantage to their survival, then they stay. But if those modifications, and they happen in the other way too, if those modifications come through and it doesn't make it better and they don't survive any better, then that variant doesn't survive. And so the environment's kind of, the pressures of the environment actually do kind of control what stuff sticks and what stuff doesn't. Everything is happening, but the changes that make a difference, that give some competitive advantage to the virus, are the ones that remain, because those changes are the ones that get transferred down to the next cell to be infected. The same model works with these genes, for instance, though. So, for instance, getting back to the dog and the sequence, the gene A is in dog and the sequence because the gene gives it some kind of competitive advantage, or rather that gene didn't cause the organism to die, to become extinct. And when something becomes extinct, just like going back to our woolly mammoth friends here, when these woolly mammoths became extinct, their DNA was lost forever, because in the crudest sense of things, the environment would say, or the environmental pressures would say, that DNA did not help this organism to survive. And so therefore, the organism died, and the DNA went away too. But they're saying, in that same kind of logic, they're saying that gene A in the dog and in the sequence here did not cause that dog and the sequence, or the organism of the sequence, to die. Now, the gene could still be faulty. It could still have something wrong with it. There's all kinds of types of organ or disable or disorders which come out of bad genes. But if those disorders don't kill the organism, then the gene will transfer. This is how viruses like AIDS, the human or HIV, human immunodeficiency virus, work. And that is that while they are dangerous and they eventually do cause some real grief and some suffering for the patient, they're not so grave that they kill the host. And when they don't kill the host, that host is able to give them on to other hosts. But this is different from, for instance, diseases or viruses like the Ebola virus, which came out in Africa some time ago, where if you got sick with Ebola, you had days to live. I mean, it's simply just days to live, and it was a very dangerous thing to have. But it kind of it contained itself, because in the days in some of those areas, the virus couldn't spread out to other communities. And again, my heart goes out to the people who suffered from Ebola. And this is one thing, on a personal note, this is one thing about why I'm so excited about bioinformatics, because in my mind, the more we know about these viruses, the more able we are to end suffering that is caused by these viruses. Another thing we're going to be talking about in this class is the comparison of proteins. Now, DNA is the genetic material that is then created, or that's used to create RNA, which is then used to create protein. And the proteins are what your hand is made out of, your hair, your face, your skin, anything in your body is made out of protein. I mean, this is different from like having a protein shake. When I say protein, I'm talking about the stuff that actually is used. It's the product. It's the stuff that's put together to create skin or something. Some organ. It's the stuff that comes together to make tissue. So, in a comparison here, you can compare two different types of proteins. So here I have what I call the healthy protein, which comes out of a gene. I have a gene, I know what gene it is, and I know that gene, when it's allowed to do work its magic here, it creates protein. Now, I have a patient who has a different type of gene, and I know what that protein looks like when that patient's protein or gene is then used to create protein. I know what the protein looks like. And so I have the genes and I have the proteins. Now, just by looking at the genes themselves, I don't necessarily have an idea about how this is going to affect my patient. However, when I have like a three-dimensional model like this, I can look at what's called a healthy protein or a wild-type protein, and then I can look at an unhealthy or non-wild-type protein. And let's just say that I'm looking at these things, I've just drawn these things in, but let's just say that I'm looking at the healthy one and I see there's one kind of big protruding green elliptical thing. Both genes or both proteins have this. So I'll think, well, if that's in the healthy protein and it's also in the unhealthy, maybe that's not the thing that's causing the grief. It's not the thing that's causing the ailment. However, when I look at the unhealthy protein, I see that down below, just like above where my cursor is now, there's that orange elliptical shape. And that's a difference. Otherwise, the proteins are exactly the same. The only difference is that one thing is in the unhealthy and not in the unhealthy, and it's not in the healthy protein. So that's the difference. And so by this kind of comparison, we can pinpoint another area of research to go into, and that is, how does that orangey elliptical thing that I see in the unhealthy protein, how could that contribute to the dishealth or the unhealth of my patient? So this is by comparison, or by doing like this comparison, you don't have the answer about what's going on, how the person is sick, but you do have an area about where you should investigate. In other words, it's like you're looking at a kind of a likely candidate for what the ailment could have been produced by. So you have diseases like, for instance, or ailments that are like cystic fibrosis, for instance, which is basically a misspelling in genes, which then create bad proteins, which then go on to create all kinds of really unfortunate disorders. And those can all be maybe brought back to this part, this exact area just above where I am right now, in the protein. We talk about that. Another thing that bioinformatics is very, very good at is creating what's called the tree of life. The tree of life. I mentioned this earlier, and that is that all life that you see is made out of the same kind of genetic material. I'll say that again. All life that you see is made out of the same genetic material, which means that you can use your same approaches that you use from the Human Genome Project to study the genes that are in a daffodil or a lilac or a rose. You can look at the same, you can use your knowledge from the Human Genome Project's gene library and the analysis, or the whole analysis, or the way that they procured the genes or got the genes out of the genome and actually wrote them down into this giant string that they call that they entered into GeneBank. That approach can be used on roses and find the rose sequence. And they've done this. Actually, most plants that are out there, you can find, in fact, seemingly all the major plants, and all the sequences of the genomes are all known. If you look at the news, you'll see that there are seemingly trillions and trillions of different types of plants out there. And little by little, they're all being sequenced. These are all research projects. In fact, there's some labs, and that's all they do. They don't analyze, but rather they just go through and pull out all the genes. They figure out what the whole sequence is. I'm kind of getting ahead of myself. I'm kind of running out of time here. I'm just too excited here. But let me just get to the next point here. But why computer science here? Because when you're working with this comparison, you're working with two different small sequences, sequence A and sequence B. It's obvious where the differences are. This is a goat. This is boat. You can see that things are actually different. Your eye can see that, and we're good at finding these differences. But when you're working with sequences that may go on for a long way, it's actually quite difficult to find those differences. And so computer science comes in to help us to find these differences between organisms so that we can create these kinds of trees and kind of find out how the differences between two different genes could be contributing to someone's dishealth. So there are some objectives in bioinformatics, and that is that, for instance, you're using, I guess the most important thing is you're using DNA to answer questions. But rather, bioinformatics is getting to the stage where we can actually answer those questions. Like, how do we design an experiment? Well, we first of all need the DNA. Okay, got it. Okay, now we need to be able to go through and quantify that DNA and find certain types of statistical anomalies in that DNA. Okay, we got it. And now we need to find a kind of a model-based system to be able to see whether those anomalies between this DNA are the same as those anomalies of another piece of DNA. Okay, you've got it. That's basically what bioinformatics is doing. It's a toolkit that allows you to do all this analysis here, to perform these analyses here, to make the data that you're finding available to other people here, to identify sequences, to find perhaps the function of those sequences here, to learn what that function is, and that's actually quite tricky to do. But basically, though, to use DNA in your analysis. And so you're using all this stuff together. You have computer science, math, and you have biology, and I put medicine in here anyway, though, because this is kind of becoming a medical science. So anyway, I wanted to ask you just you can work on this if you will. I think what I'll do is in the last 4 minutes, I'll go ahead and go into groups here. But I would like you, though, to go into a group of maybe 3 people and maybe 2 other people, and then to answer some questions on this form that you'll find down here. And then when we come back on Tuesday, I'm hoping that we can discuss some of these ideas and then kind of just kind of get an idea about what ideas do you have that bioinformatics could be used to do. So with your permission, in the last 4 minutes, I'll put you into groups, and then at 11:40, go ahead and just log out, if you will. And if you still need to discuss things with your groups, then please do. I mean, make a Google Meet with each other. But otherwise, I realize that some of us have to go on to other things, and so I don't want to keep you over time here. By the way, on Tuesday, I will be in class. You can join me online or in class if you're an in-class person on Tuesday. But I am, again, looking forward to getting to know you and your ideas and just enjoying bioinformatics with you. I think it's really exciting. It's always exciting. I'll go ahead and make these breakout rooms. I'll put 7 rooms out there. I think that'll be enough. And if there's just 2 people in a room, I'll try and move you into a group where there's 3 people. Everybody, have a good weekend. Stay safe. I'll see you on Tuesday.